Hello student, welcome to the next lecture of biological classification. Previously we discussed introduction of biological classification, few types of classification, and today after discussion of this Monera, we are going to start with the Protista. Protista is a kingdom of unicellular eukaryotes. So let's begin. Yes. So Protista, as you can see on the screen, the kingdom Protista is primarily aquatic. Primarily aquatic. Why it should be primarily aquatic? Why not terrestrial or some other? Because if you are a single cell, the water is highly essential for metabolic activities. Okay, for both food, excretion, and ingestion. Okay, so primarily the movement of a single cell organism is more possible in aquatic condition, and the life processes such as diffusion of waste and all, and primarily gases, oxygen is more possible, more feasible in aquatic environment than any other. So all unicellular eukaryotes which were not fitting in any other criteria such as kingdom Plantae or Animalia or fungi, they were put together where in kingdom Protista. Okay, so eukaryote means well-defined nucleus and membrane-bound organelles should be present. Okay, so this is the uh, kingdom whose boundaries are not well defined, whose boundaries are not well defined. Okay, okay, yes. So let's begin with the members of Protista. So Protista have Chrysophytes. So these are the photosynthetic protist, unicellular photosynthetic protist, Chrysophytes, then Dinoflagellates and Euglenoids. All these organisms contain chloroplast and photosynthetic pigments which, okay. Next, we have slime mold. Slime mold again, these are called as primitive relatives of fungi. That evolutionary fungi evolved from this. Next is protozoan, and these are also called as primitive relatives of animals. So, this kingdom Protista contains all members who are related with a respective kingdom. So these members, Chrysophytes, Dinoflagellates, Euglenoids, these are related with kingdom Plantae or plants. Slime molds are related with fungi. Okay, again, so sometimes the connection get clipped and technical glitches are going to be there. Okay, so what we are talking about is plants. These are the primitive relatives of plants, or these are plants, or slime molds, primitive relatives of fungi, and these protozoans. All these are connecting link to multicellular eukaryotic kingdoms. And one of the things we have discussed earlier here is boundaries being not well defined. So you can see here that this euglenoids, it can act as a plant but can also hunt. Okay, so it can act as a plant, it can also hunt. The slime mold can move like animalia or protozoans, and also acts like fungi. So boundaries uh, in this kingdom Protista are not well defined. They can show multiple functions of multiple groups. Next. So coming back to this, reproduction in Protista. Asexual reproduction is going to be what mitotic and all. Sexual reproduction, they fuse the cells and zygote form. Fuse the cells and zygote form. Generally the cells from other organisms, different organisms, uh, and zygote forms. Next. So if you see the rough evolutionary pathway. So you have unicellular prokaryotes here, and then we move to unicellular eukaryotes. So kingdom Protista becomes the link between unicellular prokaryote and multicellular eukaryote. Okay, so what it is a link between? It is a link between unicellular eukaryote. See, it is a unicellular eukaryote. It's a link between unicellular prokaryote and multicellular eukaryote. Okay, next. So moving to chrysophytes. Okay, our first member we are going to discuss is chrysophyte. So these are present in fresh and marine water, so no issue. And chrysophytes are of two types, desmid and diatoms. Okay, but these are general characteristic for chrysophytes. So desmid and diatom both are present in chrysophytes. So they occur in fresh and marine water. They are microscopic. This is important. Many a times a question or a statement they will ask, these are microscopic and flagella are absent. So when flagella are absent, locomotory organs are absent, they have to float passively in a water current. Float passively in a water current. Wherever the water current takes them, they will go. Next. Now coming back to the two members, one is desmid and second one is diatom. Okay, so pay attention here. Desmid and diatoms, these both or diatoms are mostly photosynthetic. Okay, so desmids are again photosynthetic. They are also called as golden algae, and pay attention here. They are also called as golden algae, and they are very good pollution indicator in fresh water. So sometimes the question comes or a statement comes, very good pollution indicator means what? Okay, pollution indicator means they grow too much where pollution is there, it's not. They actually die if the pollution is high. Okay, this is a good pollution indicator. That means if I used to see in a pond so many golden algae, suddenly over time I see that the golden algae population has reduced drastically, that means the water has become polluted. Okay, good pollution indicator means here the number will decrease, the organisms will die. Okay, next. So when we are moving to diatoms, okay, diatoms are mostly photosynthetic, they are marine, and they are chief producers in water. Chief means main. Okay, so they are chief producers in oceans. So why this is so important? Chief producers in oceans. They do the most of the photosynthesis and nearly produce, oceans nearly produce 50% of, more than 50% of oxygen on earth. So these producers are very important. And again, due to this phytoplankton, these diatoms, what happens, all the biomass created there is goes from phytoplankton to zooplankton, zooplankton to other small fishes, and small fishes to bigger fishes, and so many other animals. So chief producers in oceans is very important. Next. Next, so we have a very good economical benefit of diatoms, okay, due to their cell wall. Their diatoms, their cell wall is made up of cellulose, but it is heavily embedded with silica. So if this is a cell wall, it is heavily embedded with silica. Silica tends to embed there, and silica being indestructible, what happens after their death? The diatomous earth is formed. Okay, so the dead bodies of these diatoms accumulate, and all the inner protoplast and everything is gone. Only their shells remain. Those shells contain cellulose body wall, and embedded in them is silica particles. And those are gritty in nature. So when it is very gritty in nature and very fine, it can be used for polishing rough edges and filtration of oils and syrups. Oils and syrups. So here diagrams are there. This is a accumulated diatomous earth, which could be used in polishing or could be used in filtration of syrups. and could be used in various industries. And one of the non-NCRT uses, it can be used to make soundproof rooms. So if you have soundproof rooms, which could be used for sound experimentation, audio recording, all those stuff. Okay, so this is there. One more one more important point or keyword of this, as you can see, is two thin overlapping shells which fit together like a soap box, okay. So this is our chrysophyte, and in chrysophyte they will ask you about desmate being golden algae, being freshwater pollution good indicator, and diatoms being chief producers, and they produce diatomous earth, and these two overlapping shell fit together like a soap box. These are the keywords, and if the questions come from here, it will come from this topic. Okay, and this is a actually out of these four categories of chrysophytes and dinoflagellates and protozoans and all, this one is very important, or mostly time the question is asked from here. It is the examiner favorite. Okay. Okay, so after completion of this chrysophyte, we are going to see dinoflagellates. Again, dino, two flagellates, two flagella. Okay, so what is the difference here between chrysophytes and dinoflagellates? Dinoflagellates have flagella, and these are mostly marine and photosynthetic, and they appear yellow, green, brown, blue, red depending upon main pigment. So these are some lines, some information you have to mug it up. Okay, the key factor here, how do you remember this, is the dinoflagellates is they have two flagella, and one is here transverse flagellum and one is longitudinal flagellum. Okay, so remember this, one is transverse, one is longitudinal. And if you want to see, these are some dinoflagellates. As you can see, they have this very hard cellulose plates on them. Okay, that's why they are also called as armored dinoflagellates. They have armor, okay, to protect them from outside. They have armor. So you have reddish here gonalax, brownish sericium, green peridium, and yellowish brownish dinophysis. You don't have to remember their structure and all, just you have to remember that they contain, and this is the NCRT diagram, very simple. Which one they have given? None of this. Okay, so they have cell wall stiff cellulose plate. What they have? Stiff cellulose plates for protection, and they have two flagella. One is longitudinal and one is transverse, which is present in a furrow in between plates. So this is a furrow. So one is transverse, one is longitudinal. Furrow is like a long trench, or you can call it a channel, nali. Red dinoflagellates. When we are moving to red dinoflagellates, very economically important and healthcare wise also important significance is there. Red dinoflagellate gonalax. Remember this one. Okay, this one is favorite of NEET examiner. This is called as gonalox. So it produces a red tide phenomenon. It produces a red tide phenomena. How the red tide phenomena? When the tide comes, what happens? Overnight rapid multiplication of these dinoflagellates, entire this area of the sea coast, you can see that it becomes red due to their sheer overwhelming number, okay. And what happens due to that? The sea appears red, and which each tide appear also red. Okay, so red tide, this gonalox produces saxitoxin. Again, this is not part of NCRT or NEET, but remember saxitoxin, and this saxitoxin kills fishes. It kills fishes. Okay, and fish die by paralysis and all due to toxin. But some of them, these clams, these particular types of clams, are not affected by this saxitoxin and they can eat the gonalogs. What happens when these shellfish are eaten by humans? They undergo paralytic shellfish poisoning. They undergo paralytic shellfish poisoning. Okay, so what is happening here? This saxitoxin through this shellfish go inside of human being and human beings suffer paralysis. Okay, so this is called as paralytic shellfish poisoning. Next. So red tide is caused due to gonalogs, not gymnodium, because gymnodium not part of syllabus. This is extra one. Okay. Gonalogs 100% red tide, match the pair. Next is bioluminescence. So bioluminescence is a phenomenon in which biochemical reactions generate light. Okay, generally we see light from sunlight or from electricity. Here, uh, bodies of this uh, dinoflagellates, when they rapidly increase in number, this can be also seen, they produce bioluminescence. So if you touch the stream, uh, just like in a movie Avatar, where you can see more bioluminescence, these algae are also bioluminescent. So which algae are bioluminescent? You can take an example of Noctiluca here, leave the gonalaps. Okay, both are there in nature, but less information for you in brain to store, more information is there for to think and apply on questions. So do not fill your mind with random junk, random biology knowledge. This is a formula exam. Remember, formula exam is all about management of memory, effort, stamina, rather than random the random collection of knowledge. Okay, so do not read too much other than NCRT, but NCRT is a horrible book because of how it is created. It is not very easy to read and memorize. Okay, so you have to make notes from it. So that was maybe their intention to make reference reading and making notes for student. So this is bioluminescence shown by Noctiluca. Next. Okay, so we have completed dinoflagellates, which are mostly marine. Again, these these have two flagella. Flagella one is transverse, one is longitudinal, and they appear. Their main color depends on main pigment. Again, they have armored dinoflagellates, stiff cellulose plate. These do not forget. One is longitudinal, another is transverse, and it is between burrows or furrows. Okay. Then red tide, gonalogs, remember, and saxitoxin and PSP. This is extra good to remember. Next is bioluminescence, Noctiluca. Okay. If you really want to see how these thickle plates work, okay, this is a furrow and this is a transverse and this is a longitudinal. Simple. So this was our dinoflagellates. Moving on to next. Moving on to is euglenoids. Moving on to is what? Euglenoids. So euglenoids is fresh but stagnant. They occur in fresh water, fresh but stagnant water. Many of the students are general thing, if water is stored, it is not fresh. No. Scientific meaning of fresh water is where salt content is less compared to marine water. Okay. So they are in stagnant water and they have two flagella. One flagella is short, one flagella is long. So clear cut difference between dinoflagellate and euglenoids is, in dinoflagellates, the flagella are transverse and longitudinal, and in euglenoids, the flagella are, one is shorter while one is longer. Okay, remember this difference. cell wall is absent. This one is very important thing. Cell wall is absent, but pellicle is present. What is the pellicle? Pellicle is a protein rich layer which makes the body flexible, as it is a motile one, euglenoid. Though it is photosynthetic, it is also a motile one, and that's why here the in Protista the boundaries between kingdoms or groups is not very clear. They are merging because this single organism shows the some characters of what animal and some characters of what plants. Okay, so while it is motile, it can go on predating on smaller organism in absence of sunlight. In absence of sunlight, it can go on predating on smaller organism, that is mixotroph. Okay, in presence of sunlight it becomes photosynthetic. Again, very important question here asked, is photosynthetic and the pigments are identical to higher plants. What are the pigments of higher plants? Chlorophyll A, B, carotenoids, xanthophyll. These are the pigments of higher plants. So here we are learning about euglenoids. Okay, euglenoids. Again, if we go with what? Again, if you go with what? Thinking about that it is a freshwater organism, and but it is present in stagnant water. In stagnant water, it can actually what we call swim with the help of its flagella and capture the smaller organism, maybe bacteria, maybe other smaller amoeba or whatever unicellular organism it can grab on and eat. Okay, so this is the diagram. This is the NCRT one. So again, the uh, labels are little bit messed up. Okay, contractile vacuole is present in there, and this concept is very important. Contractile vacuole we have for salt water balance in our body, kidneys. So unicellular organisms, they form a organ, not organ but organelle. They are only single cell. So what is the function of this contractile ovule? If you can see, this contractile vacuole does what? It removes the excess water from the body. Okay, so this is a contractile vacuole. Otherwise, what will happen if fresh water, around fresh water is too much and inside fresh water is less comparatively, because other protoplasmic constituents are, all water will come inside. Without this organelle, this will swell up to some size and then burst. Okay, so we don't want that cell to die. The cell has developed to survive in fresh water independently, not parasitically. They must have contractile vacuole. They must have what? Contractile vacuole. Okay, so this is this. Many things are there, chloroplast and all, but not so important. So contractile vacuoles are important here. Again, remember, sometimes they are called as mixotrophs. Sometimes they are called as mixotroph because their trop, that is mode of nutrition, is mixed. Photosynthetic, that is autotrophic, plus they do heterotrophy by predating on smaller organisms. Okay, so moving on to next. Okay, so next topic is slime mold. Slime molds are called as primitive relatives of fungi. That means they have evolutionary relationship with fungi. They are going to show some aspects of protozoan, such as moving and capturing and eating, and dead and decaying, they are saprophytic, and some aspects of fungi, that is fruiting body and spores. Again, this is a very, see, if you study the lower organisms, they will share most of the characters. They have not yet become so concrete brown boundary fungi, animals or plant. That will occur in, that hyper specialization occurs in evolution with respect to time, okay, gradually it will become hyper specialized plant which cannot move generally, okay, animals which are totally dependent. But here what is happening, they are they are having one or two way of nutrition and one or two characteristics of kingdom. So slime mold. So body moves along decaying twigs and leaves. What is this decaying twigs and leaves? It is simple dead organic matter and it is engulfed by them. So what is the animal like feature? Body moves. Okay. Actually they show movement and they show engulfing. So engulfing movement, it is a saprophytic protist. It is what? Saprophytic protist. Then they have a particular reactions to favorable and unfavorable environment. So under favorable condition what happens, everybody is jolly, food is there, they make a group and that jolly group is called as plasmodium. Okay, everybody is friends, let's go, let's have some tours and trips. Okay, so they are having what, they are making groups. So as it looks like this, not like this, actually it looks like this. Okay, so this is a plasmodium. This is how it spreads and this is the zoomed in version. Next, what happens, always remember the question will come like this: favorable condition aggregation called as plasmodium. Okay, favorable condition aggregation, the group is called as plasmodium. Do not mistake this for malarial parasite, that is plasmodium vivax, which cause malaria. Plasmodium there, it is a name of genus. Okay, do not confuse plasmodium there, do not confuse plasmodium there. Plasmodium is a genus, here plasmodium is a under favorable condition group or aggregation of slime mold. Next, so what is happening in next? So always remember there are no definite patches of favorable and also there are no definite patches of unfavorable condition. Okay, time is what changes. So when unfavorable conditions occur, such as water stress, food scarcity, the Plasmodium, which was the earlier happy jolly groups of slime mold, it differentiates and forms fruiting bodies. It differentiates and forms fruiting bodies. It is forming fruiting bodies. Fruiting bodies is like this. Fungi produce fruiting bodies. Okay, fruiting bodies will have spores. Yeah, it is here. So slime molds will produce fructine bodies like this, and these fructine bodies produce at their tips spores. Okay, spores are unicellular propagules which have a strong cell wall and coating, and when they are dispersed from unfavorable condition, if unfavorable condition is there, you need to remain dormant. So unfavorable condition, spores can remain dormant, their metabolic rate is very low, and when the conditions again become favorable, they come out again slime mold, form colony, again they form plasmodium, again under favorable condition they enjoy. So what is happening here in spores? Spores possess true walls. Spores possess true wall. And what is mean by this true wall concept? Okay, so earlier we had this concept of cell wall, present or two kingdom classification, present plant, not present animal. Okay, then we got up to five kingdom classification and all. There what they call true walls is when the cell wall possesses cellulose, not chitin, not peptidoglycan, or not any other. So this being a protozoan, sorry, protist, saprophytic protist, it produces spores like fungus, but those spores have cellulose as a cell wall. See the how many boundaries are broken by this. First is moves like an animal, then under unfavorable condition it produces spores like in a fungus, and those spores contain cellulose in their cell wall. So truly slime molds are breaking so many molds of animal, plants, and fungi. So see here what is happening. The spores are light enough. The spores are light enough that they are dispersed by air current. They are extremely resistant to degradation or any molecules and survive for many years dispersed by air current. They are light, they are extremely resistant to biological degradation and can survive for many years. Truly, slime molds are magical organism of kingdom Protista. Okay, so hope we have cleared the slime mold. Next one is going to be protozoan. Next one is going to be protozoa. Okay, I think we should stop here as 30 minutes are over, nearly 31 second is there. So what we have studied today is we have started from Chrysophytes or the kingdom Protista. Then we went to Chrysophytes, we saw what is important there. And again, I would like to mark microscopic flagella absent float passively, and there's a diatomous earth and desmids being good pollution indicator. These are the points where they ask question. And soap box. Next, uh, here in dinoflagellates, important thing is the flagellum. Okay, transverse flagellum and longitudinal flagellum, and this gonalox, stiff cellulose plates. These are the important things. Red tide. Euglenoids, generally this mixotropic mode of nutrition, sunlight absent, becomes predator, sunlight is present, photosynthesizes, it's all and all good. So cell wall is absent, pellicle is present, so that is there. Fresh but stagnant water. Fresh but stagnant water. Slime mold we studied in detail. So much of interest that primitive relatives of fungi, their spore cell wall composition is important. What they do in favorable conditions, what they do in unfavorable conditions. Okay, all those things are very important. Next, their fruiting bodies, spore wall. So I think this much content is enough. Thank you. Okay, so study this by making notes and study in terms of questions. What kind of questions can be formed from this data? If you study by this reverse engineering method, that study for the question. Okay, what kind of question can be framed? If you think like this in systems and questions, the biological study becomes easy. At the end you have to mug it up. Okay, mugging up, keeping it in memory is very important. That is a very basic skill. Level 1 DPP questions are solved by it completely. Those are relief questions when they come in NEET. Okay, because they increase your speed. But the current trend of NEET that is happening is not the MCQ type, that memory based, but they are going with the statements and statements and statements. They are mixing up statements, they are changing very small things, they are creating traps. That for that analysis along with memory, your analytical logical reasoning should be very strong. By that way you can identify what is not being given there. Okay, what is being wrong. Okay, so this will come from deep study, and deep study is required. And read the NCRT many times and try to make your own notes or copy my notes, or try to, rather than making notes, try to make your own question, match the pairs and all. Make the, if there's so much of information is there, create graphs, make match the pair, and slowly you'll try to realize, understand the language of NCRT and how they are framing. Once you understand this, uh, the work becomes very simple. And yeah, that's about it. That's about today. Okay, so meet you in next lecture. We will continue the protozoans. These protozoans have four categories, four major groups: amoeboid, flagellated, ciliated, and sporozoan. So all these four we will discuss with one single lecture. Thank you. Happy learning.